everyone, and thank you for attending today's webinar, which focuses on product development using the NDA 505(b)(2) pathway. This webinar is co-sponsored by Camargo Pharmaceutical Services and Capsugel Dosage Form Solutions. Besides this webinar, Camargo and Capsugel are collaborating on designing and delivering a number of B2 forums. For example, GPHA's Fall Technical Conference and CPHI this year. Capsugel also joined Camargo's annual workshop in Israel earlier this year. My name is Jim Coward. I head up the market development activity here at Capsugel Dosage Form Solutions, and I will be moderating today and providing a short overview of the Dosage Form Solutions business unit as an introduction. We should have 5 to 10 minutes at the end of today's webinar to take questions. Any questions, please submit at the lower portion of your screen, and we will either answer those during the webinar at the end or via email after the event. Let me first provide an introduction to our speakers. Ken Phelps is President and CEO of Camargo Pharmaceutical Services, which he co-founded in 2003. Camargo is the most experienced global strategist on the 505(b)(2) approval pathway, which is its sole focus, and partners with companies around the world in assessing B2 product opportunities and building and executing product development plans that, based on their experience, is fully aligned with FDA guidelines. Camargo's team has successfully guided hundreds of FDA approvals. Ken will provide an overview of the B2 pathway as well as an overview of Camargo's processes. Trevor Weigel heads up the product development team at Capsugel Dosage Form Solutions, having joined Bend Research in 2003. A large percentage of our product development is with clients pursuing the B2 approval pathway. For example, improved formulations based on enhanced bioavailability or improved site delivery, pediatric applications, fixed dose combinations, etc. We have approval to present a couple of representative case studies today, and Trevor will discuss the approaches taken to meet these products' target product profiles. Trevor is a professional engineer and holds an MS in mechanical engineering from Stanford University. And Bend Research follows a specialized model for the integrated design, development, and production of innovative dosage forms for our clients. We tend to focus on specialized applications such as bioavailability enhancement, modified release, high-potency APIs and controlled substances, pediatric and orphan applications, and many of these require tailored processing techniques. Let me touch briefly on the core pillars that make up our model. Science and engineering are our core strengths. We develop a fundamental understanding of the client problem statement, and our definition of the problem statement is at times different than what our customer may have originally defined. We feel that only through a deep understanding of the physicochemistry of the API and biological properties of the site where the compound is to be administered can an optimal approach and process for meeting a customer's goals be effectively designed and developed. We do not promote a specific technology or finished dosage form presentation, but instead work with clients to develop an optimized approach in meeting their target product profile and commercial objectives. This type of unbiased approach is possible at Capsugel because we have breadth and depth of technologies and have invested to ensure a deep know-how across solid oral dose processing techniques. We factor in ultimate manufacturability and incorporate QBD principles at the early stages of product development, and our experience includes the advancement of more than 1,000 compounds. Another core pillar is our product development teams, which are well-versed in the fundamental sciences, material scientists, mathematicians, physicists, biologists, chemical and mechanical engineers. These teams tend to complement the applied science expertise of our customers, and the team makeup is tailored to the specific client needs and project phase. Because of our approach and the trust it builds, we tend to be looked upon as an extension of our clients' product development efforts versus being seen as a contract vendor. We continue investing in R&D and infrastructure at our co-located product development and manufacturing facilities in the U.S., UK, and France, and just recently brought online the largest integrated spray-dry dispersion process in North America. Slide 4 provides an overview of the technologies we utilize in product development, and this toolkit is well suited to meet typical 505(b)(2) product objectives. We have a full range of both bioavailability enhancement technologies and expertise, and modified release solutions, both in controlled release, delayed release applications to meet client compound target product profiles. This includes a deep expertise in amorphous solid dispersion approaches, both spray-dried dispersion and hot melt extrusion, as well as lipid-based formulations utilizing liquid fill hard capsules, soft gels, or lipid multi-particulate technology based on melt spray congeal processing, as well as nanomilling and nanocrystallization. Our modified release range includes new intrinsically enteric capsule technology and proprietary osmotic approaches for zero-order release, both fluid bed layered multi-particulates as well as matrix multi-particulates inclusive of lipid multi-particulates and mini-tabs, and these approaches are all routinely utilized in pediatric formulations. Colonic delivery approaches, leveraging liquid-filled hard capsule technology, fixed-dose combinations using both bi- and tri-layer matrices and multi-particulate approaches, inclusive of capsule and capsule technology. We're also well-versed with taste masking approaches, critical in pediatric formulations, and our particle engineering expertise is leveraged for pulmonary delivery and has been used in helping clients convert delivery mode from parenteral to pulmonary. Now we'll turn the program over to Ken to discuss the 505(b)(2) approval pathway.
Kind introduction there and the background on Capsugel's technologies, and we're going to be talking about those kinds of technologies, and later on Trevor's going to give some examples, some case studies of those technologies that have been applied to actual products. And in my presentation, I'm also going to talk about some other applications of different kinds of technologies and different sources of 505(b)(2) products. So, also going to, first of all, start off with the drug regulatory pathways. I understand there's, gosh, pleased to hear over 500 of you are on this webinar today. And I know many of you have experience in 505(b)(2), but some of you do not. So that we're all in the same understanding of what a 505(b)(2) is, I'm going to ask your indulgence for those of you who know about 505(b)(2) when I go through the regulatory history. And then I think all of us can benefit from learning about other sources of these drug products. And lastly, I'm going to cover development process as we've been using it successfully at Camargo now for almost 13 years. As a basis, I'd like to point out that the 505(b)(2) comprises one of the three possible ways that you can get a small molecule through the FDA. You notice I emphasize small molecule. You're all aware of biologics and biosimilars. Those go through a different process at the FDA. So probably the simplest way to get a drug approved at the FDA is through the 505J process. That's the ANDA, or the generic. These are drug products that are exactly the same as the approved products. And when I say same, I'm not just talking about the physical product. I'm also talking about labeling. Labeling is key to differentiating between a generic product and a 505(b)(2). The other extreme would be the 505(b)(1) NDA, and this is where the large pharma companies, your Merck, your Pfizer's, etc., where they conduct all of the research from discovery all the way through post-approval. These applications contain investigations that have been completely paid for by the sponsor or on behalf of the sponsor. So we tend to think of a 505(b)(2) as somewhere between, or a hybrid between an ANDA and a full NDA. Specifically, a 505(b)(2) is one that relies on investigations that were conducted by someone other than the sponsor. We typically think of these other applications or investigations as being from the public domain. I would like to point out something that's really important on this page. By now you've mostly read this thing, but I want to make it very clear that a 505(b)(2) does not have to rely on a drug that's been approved in the United States before. This is something different than a 505J or a generic. A generic, the product that's copied, is one that's been approved in the United States. A 505(b)(2) does not require that the drug that you're relying on or drug that you're going to be using is approved in the United States. So the information that you're using for a 505(b)(2) does not have to be based on a drug that's already approved in the United States. And we're going to be talking about how that works out for sponsors for sources of products in a few moments. 505(b)(2) is just exploding. As you can see, that since 2004, we've had over 350 505(b)(2) NDA approvals, and we're really proud to have been involved in about 20% of these approvals in the last few years. We expect that this number is going to continue, and I'll talk to you about why that's going to happen in just a moment. One of the reasons, though, that 505(b)(2) is becoming a very popular application process and development process is that it has lower risk, lower cost, and it's faster to get these products to market than certainly a 505(b)(1), which has enormous costs, faces enormous risks, and takes up to 10, 12 years to get to market. But we've seen examples where 505(b)(2)s can be put together and approved in less time than a generic. I'll admit that the market opportunities will be different, but in many cases, the market opportunity for a 505(b)(2) is much greater than a similar type of product or process in 505J. Again, bear with me if you're already familiar with the laws in the United States with respect to food and drugs. But I want to go just through the origin, because the origin of the food and drug laws will show you some opportunities that you may not have thought of for coming up with new product ideas. In 1906 is when we first had laws governing drugs, and we set some standards for strength and purity, and for the first time required ingredients to be listed. But it wasn't until 1938 when we had a problem with some people dying from sulfanilamide that contained ethylene glycol, which is antifreeze, that the United States Congress passed the Food and Drug and Cosmetic Act, as more or less as we know it today, that required scientific evidence of drug safety prior to marketing. Maybe surprisingly, though, it wasn't until 1962 that the laws were amended to require that drugs also work. That is, they had to be shown to be efficacious prior to marketing. Notice that the quite a time difference between 1962 and 1938 and 1906. I'll ask you to think about, what about the products that were on the market in 1938 through 1962? What happened with those kinds of products? Well, it turns out that just one moment here. There we go. That in 1984, Congress decided to do something about basically three problems. Two of them are very well known. The one problem was that large pharma had issues with getting their products approved in a timely manner to preserve the patents that they had applied for. In some instances, there were cases where the product was approved about the same time the patent expired. And that did not provide enough incentive for large pharma to invest the multiple millions of dollars required to get approval. So the effort was for them to get a patent extension, patent term restoration, if you will. The other problem is we had no defined process for generics. So there was a need to have this compromise in 1984, which said that we'd allow the entry of generics, but at the same time we would extend the patent term. So this is what we call the grand compromise for 505B1s and 505J. 505B2 was a solution to another problem, and that problem was what was called paper NDAs. These were NDAs before 1984 that were approved based solely on literature. Now, 505B2 is a bit different than that, in respect that sometimes in a 505B2 we have to do additional studies. But prior to 1984, the approvals were based upon paper only. Paper meaning public information, publications, and so forth. So 505B2 was promulgated in 1984. We didn't have proposed regulations till 1989, and people still didn't understand them. The FDA didn't think very highly of their own regulations. So it wasn't until 1992 that we actually have regulations. And furthermore, lastly, it wasn't until 1999 that we even had draft guidances. And between 1984 and 1999, I think we had something like 16 505B2s approved, hardly used. And I'm proud to say that I was involved with a couple of those 505B2s that were approved during those times. So I'm going to return back to that history now and suggest that one of the opportunities that we have in 505B2 is to take what are so-called Desi products and get them approved. Desi is a process that the Congress made back in 1962 to try to understand whether products made prior to 1962 were efficacious. Remember, 1962 was the first time that drugs were required to be shown efficacious. So they made a contract with the National Institutes of Health to review the drugs that were on the market prior to 1962. Unfortunately, it was a large program. It wasn't very well funded, and most of the products that were to be reviewed did not get reviewed. And so at this time, we have more than 3,000 products on the market without an FDA approval. Some of these drugs are very well known and have very large markets and are still unapproved. And my thesis is that getting these drugs approved presents a very good commercial opportunity. We're pleased to say that just 2 weeks ago we filed, I think, the 4th NDA for a Desi product for one of our clients. We've filed numerous NDAs for Desi Products this year and past years very successfully, often with very little additional clinical studies to demonstrate safety and efficacy. We can get a lot of that from the public domain. This next slide, I'm on slide 14 of your slide deck, are some examples of Desi products that have been approved to the 505(b)(2) approval process. We have the pancreatic enzymes, codeine, certainly well-known products, colchicine, morphine, oxycodone, and so forth. These are all have been approved through the 505(b)(2) process, and that is the process to get Desi products approved. What I'd like to do in the next few slides is show some other areas that we can explore for product ideas to develop through the 505(b)(2) process. For many people, it seems surprising, but 505(b)(2) can be used to get new chemical entities approved. But when you think about it, go back to my definition or back to the law that I mentioned earlier. 505(b)(2) does not depend upon a currently approved drug. So, for example, the Desis, those are all unapproved drugs. Other examples of drugs can be from outside the United States, or in this particular case, in this list, you can see, for example, caffeine citrate. I'm sure many of you are watching this webinar drinking coffee or drinking your Coca-Cola products. And the caffeine and citrate in that product or in those drinks is the basis of a new chemical entity. There's tremendous advantages to getting new chemical entities approved through 505(b)(2). These are just some of the examples. And again, we're so pleased to be involved in the approval of some of these products going forward. It's also possible to get new indications for products. Generally speaking, new indications are for products that have been approved in the United States. However, we have numerous examples where we've taken drugs that have been approved outside the United States and add indications or find new indications and bring those products into the United States. So this new indications does not depend upon a currently approved drug in the United States. Here is a list of examples of new indications. It's also important when you're thinking about new indications that you simply don't add the indication to an existing generic product, but there'll be new routes of administration, for example, on this slide, or new dosage strengths, new dosage forms, new routes of administration, as I already mentioned. Another area that's very popular, and I think increasing the popularity, is the use of what are called prodrugs. And this is where the parent drug, I call them the parent drug, but that's the drug we already know about. Again, that drug does not need to be an approved drug in the United States. It could be an unapproved product, or it could be a product from outside the United States. But this parent drug, to this parent drug is added some sort of moiety. In this particular example, it's a lipid. And the way in which this bond is formed between the parent drug and the lipid gives rise to a new chemical entity. And again, that has great advantages in terms of exclusivity and protection from generic competition. The question about prodrugs and the degree to which you have to do studies depends almost entirely on when the prodrug becomes this reference listed drug in this case, or the drug that's already known. Drugs that are cleaved in the stomach or in the intestine don't require huge additional studies, whereas drugs that don't cleave until they get into the tissue require probably a very large number of studies. But prodrugs can be a very popular means of getting a product approved through the 505(b)(2) process. Racemates have been popular. Again, we've had in the past we've had racemates, and some people are taking the isomers and finding out that the isomers have greater efficacy or safety. We're not limited to the active ingredient. In this particular case, this example, an excipient change made for a better product. And we're seeing quite a few excipients being used to modify existing products, again, U.S. or ex-U.S. products. These excipients are used to change the route of administration, to improve the delivery, and certainly I think Trevor's going to be talking about a couple of examples, but this is where Capsugel excels in terms of changing excipients to provide better delivery for safety and or efficacy. On slide 20 at this point, dosage form changes. Here again, this is undertaken to provide better efficacy or better safety. In this particular case, we have a sublingual or lingual spray, we have gels, inhalation. The last one also changes the indication. Fixed dose combinations are quite popular. There's quite a number of commercial reasons as well as medical reasons to do this. In this particular example, I'm trying to point out a couple things. One is that the Zyrtex, they added pseudoephedrine in 2001. The way to get the pseudoephedrine added to Zyrtex was through 505(b)(2). The D stands for pseudoephedrine. I think most of you would understand that pseudoephedrine is in the public domain. Nobody developed pseudoephedrine. There's no NDA for pseudoephedrine. And the 24-hour release was done from taking an original NDA through OTC. And 505(b)(2) is the route to get an NDA product through to an OTC product, and we're pleased to, again, to have been involved with some of the OTC switches, if you will, in the past. Parenterals are very popular projects for us. Again, changing excipients, changing the presentation. 505(b)(2) is the way to go, because as most of you are aware, you cannot make changes for parenterals, even in the excipients, to an extent through the generic process. You have to use 505(b)(2). Pharmacokinetic alteration, again, Sarah's going to provide some examples, but this is basically improving bioavailability, to target the release, changing immediate release to extended release, and to reduce the variability of products. Some products have very high peaks and very low troughs, and those cause problems in the patients that can be remedied by pharmacokinetic alterations. Here was an example where intermezzo, where the product was designed to provide some additional drug levels during the middle of the night to prevent people from having to wake up in the middle of the night. Example of PK delay. Again, while you're reading the slide, basically I'm pointing out that what we're trying to do is to improve the coverage of a drug throughout the day to provide the correct amount of drug when it's necessary. Here's an example of PK improvement. This is propofol. Here you can see the goal here was to reduce the big initial spike, which, as many of you are aware, propofol is the so-called Michael Jackson drug, and Michael Jackson is purportedly an example of the effects of this high Cmax causing death. Example of pharmacokinetic alteration of IR to ER, again, trying to reduce the frequency of dosing, which also can improve the safety profile, in this case reducing gastrointestinal side effects. Before I switch this over to Trevor, I'd just like to give a couple of moments to talk about the process we use at Camargo, successfully applied to dozens and dozens of 505B2s. The process is distinctly different than a B1. If you know about the B1 process, I would encourage you to look at our blog to give some direction as to how it's been modified for 505B2. And for the folks that are involved in the generic 505J, this would be totally different to you. But I would emphasize that when you're starting off looking at a product, the first thing you need to consider is whether it's feasible. It may look possible. For example, you can improve the bioavailability, but it may not be medically important, may not lead to a differentiated product from a commercial point of view, and or it may not be possible to get regulatory approval. And we really stress the latter part of that, is getting regulatory approval. I mentioned very early in my talk here that the important part of 505B2 is to get the correct wording on the label, because that can lead to either commercial success or a mediocre product or worse in the market. So the important thing early on is to determine whether it's feasible. As I mentioned, you need to know the medical, the scientific, the regulatory, and the commercial aspects of the product. All four of these aspects need to be established prior to embarking on spending much money for a 505B2, and these are the core strengths of Camargo, is to evaluate those four things. Next after the feasibility is the pre-IND meeting. And we don't have time to go through what a pre-IND meeting is, but basically it's the laying out to the FDA of how the NDA is going to look at the end for approval. All of the constituents of an NDA coming from the public domain, augmented by whatever you need to do. That so-called gap that's supplied by sponsored studies. And this lays out the roadmap to the FDA for 505B2. Alterations as you go through the IND, phase 1, phase 2, and phase 3, if you make any changes to that roadmap, you need to make sure that the FDA is completely on board with those changes in order to have a successful NDA. We have many examples where we've reached the point, where a client has reached the point of submitting to an NDA, and the changes made during the development have caused the FDA to refuse to file that NDA because it didn't understand how that fit into the original agreement at the pre-IND stage. So this is critical. Camargo is at the FDA 4, 5, 6 times, I think in October we're at 7 times doing pre-IND meetings at the FDA. There's no substitute for experience at that pre-IND meeting and subsequent management of the regulatory package through NDA approval. I can't stress that enough. We're also pleased that because of this knowledge of the FDA and the number of products that we've been involved with, that people come to us seeking ideas for new products. And this cartoon illustrates the basic process, is taking the world of all existing products, looking at those needs for improvements, looking at the commercial, regulatory, medical, and scientific aspects, and coming up with opportunities that result in differentiated products with high return on investment. We're pleased to be involved in several of these kinds of projects and look forward to doing more. But enough of me, enough of Camargo. I'd like to turn this over to Trevor, who's going to talk about some additional case studies that are utilizing the 505(b)(2) approval process, but making a difference in patient lives.
Thanks, Ken. As Ken, thank you for going through the regulatory side of this, and another key aspect to delivering a 505(b)(2) product is the actual product itself. And so how are we going to formulate it? How are we going to manufacture it? And as Jim pointed out, and Ken both, modifying a PK is critical in a lot of the 505(b)(2) applications, and drug delivery can play a key role in delivering that modified PK. The goal is to change the performance, indication, efficacy, or other functional form of a product. I have a couple of case studies we'll go through here today. These case studies are focusing on multi-particulate technologies. That's one platform technology that we apply regularly here at Capsugel Dosage Form Solutions. And so this is where the multi-particulate technology can really provide an advantage over what the original dosage form was that came to market as the original NDA. So azithromycin, it's been long known through IV dosing of azithromycin that bolus dosing of azithromycin can be efficacious to a broader range of indications. In addition to being more broadly applicable, it also partitions into fatty tissues and creates a long sustained life within the body. So the real problem statement with azithromycin is, how do I get a large bolus dose on board orally so you don't have to have intravenous dosing to be able to handle some of the bacterial infections that it's designed to treat? This is a fairly unique problem statement because of the high dose. So it's a 2-gram dose, and if not dosed properly, emesis will result. So we have to not only not have it get absorbed too fast, but then we have to get it absorbed fast enough to where it doesn't get into the lower GI where it's not absorbed at all. So the high dose and the narrow target window make azithromycin a difficult but worthwhile 505(b)(2) product to look at. That's basically the background for the formulation of the target product profile that became that we went after. So if you look at the actual product goals themselves, we had the combination of release rate, dose, and dosing route that led us towards this multi-particulate platform. The release rate, when we look at a controlled release product, we typically start with the geometry, and we think about what is the form that's going to give us a release rate that's relevant for this particular target product profile. And in this case, we looked and said, well, with a multi-particulate in that 1 to 500 micron particle range, we believe we can achieve that release rate target that we're looking for. And when we said, okay, well, it needs to have a 2-gram dose, well, that immediately drops it into a sachet if you're going to try to have a single dosage form rather than a whole pile of tablets or alternative approach. And when you're going to have a sachet of multi-particulates, that requires good mouthfeel. And so multi-particulates, they can have a gritty, sandy taste in your mouth if their particles are too large. And so we designed these particles particularly to be smaller than 250 microns to actually not have any mouthfeel, as well as excipient choices to have a very slippery feel to them so that you don't feel them at all in your mouth. As with any formulation, we need to enable the performance, manufacture, and stability. This particular compound had some interesting stability challenges, both in terms of maintaining polymorph as well as the functional performance of the multi-particulate itself. And it's balancing all of those that gives you a good product. So before I talk in more detail about the product itself, let me talk a little bit about the technology that is enabled. So the physical situation of the actual particles themselves is an interesting technology because what we do is we create a homogeneous mixture of both a non-water-soluble matrix polymer as well as a pore-forming matrix material, and then finally the water-soluble API. And the basic functional form is that this particle, upon in vivo release, will start to dissolve the pore former and create tortuous channels that connect all of the crystalline API to the surface of the non-water soluble core. And through that mechanism, we can actually control the release rate. It's a fairly broadly applicable technology, and it requires an understanding of the physical chemistry to make sure that the pore networks connect all the way to the surface. But with this technology, we've been able to formulate many drugs on this platform. So, as is usually the case, the performance of this particular formulation to meet that target product profile, you have both formulation and process that you can modify to be able to hit your target. And typically what we're trying to do is optimize the target product profile as well as the performance and manufacturability and stability all at the same time. And with this platform technology, we were able to both adjust the ratio of the excipient, so in that case we were adjusting the water-soluble pore former to basically create more pores and or larger pores to decrease the tortuosity and increase the flow rate out of the particle. And then on the process side of things, the surface area of the particles themselves have a major impact on the release rate of the drug. And so we were able to basically tune in our below 250 micron target product profile range to also adjust for release rate. So with that fundamental understanding of what it is that drives the performance of it, we were able to formulate and then design a process into a robust space that enabled repeatable and reliable manufacture. So in this particular case, the LMPs were the initial target, and that was primarily based on the sachet, the high dose, as well as the target release concept. And from the performance standpoint, we had to hit that narrow window of target, which we were able to achieve with this single unit operation. The good mouthfeel and patient acceptability, sachets are something that different markets have more acceptance of than others. In this case, the compliance side of a single dose of azithromycin is actually a really key factor in enabling this to be a good product profile. And the science and engineering that we applied to this to get the formulation in a very narrow but controllable space, as well as the process designed, we fell back to the fundamentals and ended up with a very robust process. And currently this product is approved for US and Japan use. So on our next case study, I'm going to talk about another multi-particulate. This one is for case masking for pediatrics. The case masking for pediatrics is a critical area of application which a lot of our customers are very motivated to work in these days. Basically, you have an API with a bitter taste, and for us parents out there that have struggled with administering medicine to our kids, this is a very well-known and familiar problem statement. I've been fortunate enough for most of my dosing to my kids to be the cure of a runny nose or maybe the occasional ear infection. But when the child has a life-threatening illness, dosing of a potential cure is more than a convenience, and that's where these types of technologies are really critical. So the example here, what we have is actually an oncology drug that I'm going to go through. And the kids were literally rejecting the administration of their life-curing medicine because of the fact that the taste was so bitter. And when you're talking to a 3 or a 4-year-old, that's a difficult conversation to have, and you can imagine how emotional a parent would be at that stage. So it's truly enabling a better cure. So the target product profile on this particular application was an oncology drug, as I mentioned. It's a small molecule. We have a dosage form that needed to fit into a size 0, size 3 sprinkle capsule that would be then poured onto a dosing vehicle. We needed the small particle size for mouthfeel, as I described before. And this one particularly was not a large dose. The sort of dosing regimen per meter squared there is shown. And the intent was to be able to sprinkle this onto a food or into a liquid media, and potentially even take the capsule if the child gets old enough. We needed to have this particular release rate show bioequivalency with the IR adult formulation. That was a critical part of this 505(b)(2), and it obviously needed to be a robust manufacturing process with excipients that were acceptable for the pediatric domain. In this particular case, with the target product profile outlined above, we started with an IR core that was manufactured in the same fashion as the melt, spray, congeal process. The excipients were obviously significantly different to not enable a controlled release, but to actually enable an IR MSC core. And with that, we took that core and we evaluated 3 coatings, 2 reverse enteric options and 1 erosional controlled options. These 3 prototypes were evaluated, were initially chosen because of the TPP and then really evaluated in vitro against those TPP targets. To simulate the in vivo performance, the in vitro evaluation of these prototypes require both a taste mass test as well as a release test. And you can see here the general test that we performed in vitro. We initially had a release test in simulated saliva fluid, where we had a target we were trying to hit below 5% released, and I believe it was 10 minutes was the time. And then once it was transferred from there into a gastric buffer media, we needed to have immediate release all the way to 100%. The next slide here I show the actual results measured up against the actual criteria of success. And if you look at the Operdry, which is an erosional coating on the outside, it basically gave us a controlled release effect, which was acceptable in some cases where you have a low bitter taste threshold. That could be an acceptable output, but in this case, with a very tight target and a very low desired release rate, that was too fast of a release rate to be able to hit our 10-minute 5% release window. And it did enable actually a little too slow of release also for the IR stomach release to hit our bioequivalency. But on the reverse enteric, which the concept there is that it requires a low pH to be able to dissolve the coating, and when it does hit low pH, it dissolves very rapidly. And both of our 10 and 20% reverse enteric coatings showed very good promise to be able to hit not only the lack of a release in the simulated saliva, but then finally the actual release rate of 100% to get as our bioequivalency. So the key point here on the taste masking side of things is our clients, they have very good understanding for their molecules, their patient population, what they're targeting, and also what will be a successful product ultimately. And we take our science and engineering expertise as well as our technology platforms and combine those together with our clients to make better products. Ultimately, this needs to be something that's going to be palatable in the clinical trial to children of the ages of anywhere from infants all the way up. And that's a very difficult target to hit. But with this platform technology, we've been able to deliver many medicines that way and show very, very good in vivo efficacy in the clinic. So to wrap up, what the 505(b)(2) product development summary, basically with the 505(b)(2) regulatory pathway, our broad toolkit of technologies that Jim outlined earlier, both whether it's talking about solubilization or modified release, requires some pretty significant technology. To apply these technologies, it takes the science and engineering, which is where our core really lies. The multi-particulate platform is a flexible format that allows you to hit several of those targets. And together with our clients, we can take our engineering and scientific capabilities to create new products that create an improved efficacy and, as I think Ken pointed out at the end of his, better products. And that's ultimately the goal that we're looking for, is really to make better products for our clients and for our patients. With that, I'll turn it back over to you, Jim, to answer any questions.
Okay, thanks, Ken and Trevor, for a very informative discussion. We now have some time to take some questions. As I said earlier, we will answer all the questions submitted either by Camargo or Capsugel Dosage Form Solutions. We'll take a few of those live now. Okay, we have several questions have come in. The first question is more of a technical nature. Will the slides be provided? We will provide the slide deck to the participants today. If you've registered or attending here, you will receive a slide deck from us. And again, we'll follow up questions that we don't answer now in the coming days via email or phone call follow-up. There's several questions that have come in. The first few are for Ken. Ken, I just read the questions off to you. We have one. Let me get to, if the patent of parent drug is still effective, can its prodrug file as a 505(b)(2)?
Yeah, thanks, Jim. So we're talking about is prodrugs, and the question is if the parent drug still is under patent, can you file a prodrug? And the answer to that question is no, because when you're, or at least I'm presuming it is, because I think the question deals with the parent of a drug being patented under the U.S. patent law, which therefore means it's been approved in the United States. So therefore, you're going to be using that parent drug as one of the listed drugs for your prodrug. So as long as the listed drug is under patent, you cannot file 505(b)(2) unless the patent will run out prior to your approval.
Okay. Thank you. Another question for you, Ken. Can you talk about regulatory exclusivity possible with the various types of 505(b)(2)s?
Yeah. So currently the major types are no exclusivity at all. You can gain 3 years. It's possible to gain 3 years for when there's a requirement for a clinical study. And a clinical study is defined as phase 2 or phase 3, not phase 1. So if a clinical study is required for approval, you can potentially get 3 years of exclusivity. 5 years exclusivity is gained when you have a new chemical entity. I mentioned in my session about the new chemical entities being approved under 505(b)(2). And lastly, 7 years for orphan drug designated products.
Another one for you, Ken, assuming target for securing the same indication as listed drug via the 505(b)(2), what extent of clinical development may be required?
You know, the question here is a relatively involved question. That was the portion of the question. The participant is really asking what types of 505(b)(2) developments can be done strictly with just a BE type of study. In other words, relative BA using the traditional 80 to 125% confidence interval that's traditionally assigned to a generic. Clearly, there's many cases where a pharmacokinetic type study can be used for approval. Generally speaking, a pharmacokinetic study is very often needed in a 505(b)(2) simply to bridge between the known safety and your current product. We don't have time to go into bridging, but the bridging is a very important concept in 505(b)(2). The need for further clinical studies, phase 2 and phase 3, depends upon basically pharmacokinetic differences and pharmacodynamic differences. Particularly, for example, if you change the route of administration, you need to look at the impact on that particular route to determine whether or not the toxicology has been firmly established in the listed drug or in the literature. And if not, you may end up doing some additional studies. Also, route administration may affect the pharmacokinetics, and therefore you may have to adjust the dose, which then require phase 2, and then ultimately, depending upon the indication, you may end up having to do a phase 3 trial. But it's what the devil's in the details for 505(b)(2, and hard to give a blanket answer to that question.
And I would thank you, Ken, and I would encourage some of the listeners, if you do have detailed questions, you've got the contact information here on your screen for follow-up, and I'm sure the Camargo team would be pleased to answer it in more detail to your specifics. We have one question here for you, Trevor, on LNP technology. Can you comment on drug loading possible with lipid multi-particulate technology, and how does it compare with fluid bed processing?
Yeah, sure. So the basics are that it's going to be very, very molecule dependent. I think most folks on the phone are going to have an understanding of that. It's going to be molecule and target profile dependent. But just as a frame of reference, since that's probably more the type of question that it actually is what types of loadings have we been able to achieve with water-soluble drugs? And for some of the controlled release with water-soluble drugs, we've been able to achieve significantly higher than 50% loading in using this LNP technology. And if we're trying to go for an IR core, we've also been able to even push that significantly higher than that, closer to the 80+%. And as far as the fluid bed processing side of it goes, and how does it compare, you can achieve really high loadings with fluid bed processing as well, and a lot of it depends on if you're going to start with a neutral core, some sort of sugar core, or a inert core, and then coat your way up to high drug loading. The one thing about fluid bed processing is that coating time can get pretty long when you're trying to end up with a high overall drug loading, but it's also an achievable space. And again, the choice between going LNP versus fluid bed has more to do with the target product profile and the actual active that you're using.
Okay, thanks, Trevor. We've got time for one more question. There's a lot of questions coming in. Ken, this last one will be for you. Can you comment on recent FDA guidelines? It would appear that the FDA is increasingly facilitating the B2 approval pathway.
Yeah, I'm not. The FDA is refining the entire process for 505(b)(2) approvals. They've learned a lot. Industry's learned a lot. Folks like us have learned a lot as well. The depending upon the division, and it's very, very division dependent. I can't stress that enough. We'd like to think that all divisions are approaching this the same, and they are absolutely not. Depending on the division, I think the most significant changes can be looked at in terms of the understanding, the new science around toxicology, the impact of the adverse events of more recent drugs in terms of, for example, the cardiovascular impacts and the need to do additional studies over that of the original, the approved drug. And lastly, I think, and there's many other things, but I would comment that the process of going from the pre-IND to the NDA has become stricter, under greater scrutiny, to make sure that everything that was agreed upon earlier is incorporated in the NDA. We've seen other people coming to us that found that to be difficult and not been able to do it. So we've seen a lot of rebounds that would come our way to be fixed. I can't stress that enough.
Okay. Thank you very much, Ken. With that, we're over the 60-minute mark. Thanks again to Ken and Trevor for an informative discussion today. We will follow up as discussed, and you have the contact information on your screen should you need more detailed follow-up. Again, thank you very much for attending. Have a great day.